I'm going to introduce Sam Barrons to his topic today, which is DNA isolation and bacterial diversity analysis from cave soil to soil. Okay, and I'll just mention this. Sam's been working in my lab for about 2 years. 2007. And he really, what impressed me with Sam was that he came to me with a project already in mind. I didn't have to really direct him on some little side project that I already had in mind. He came to me with an idea. His idea was studying microbes from cave soils. And number one, I'm not a microbiologist. And number two, I don't do caves. I'm claustrophobic, so I prefer not to go into caves. But as he will attest, I went along with him for sample collection anyway. So I was that was fun, but a one-time deal for me. So anyway, and the other thing is Sam was interested in studying microbes from soil samples, and there's nothing really dirtier in terms of trying to isolate enzymatic quality DNA than from the soil samples. So that Sam will attest to, and he will mention, was rather difficult. He has one slide that describes it, and we spent months working on techniques. In fact, I called one of my colleagues and friends that are really eminent soil scientists, especially from soil samples and dealing with microbes from Antarctica. And when I asked him, what's the best way to isolate nice, clean DNA samples from soil, the first thing I heard on the other end of the phone was laughter, and then trial and error was the other answer he gave. But as Sam mentioned, there was some stumbling blocks there, but we got past those. And he's really become a sequencing expert, and I hate to see him graduate. So with that, I will leave it with Sam, and let's do it.
All right, like I said, this has been my project for my thesis here, and this is a thing that really interested me was caves in general. And when I started getting into some of Dr. Blair's classes talking about microbiology and microbial ecology, it really made me think about how these environments are pretty unique, and I'll go into a lot of that. So today I'm going to talk about a lot of the main things that we went through in this project, and I'll start off with talking about the purpose. So the purpose of this project was really to look at the habitat and look at trying to set a baseline for the habitat as a preservation method. And if we know what's naturally occurring in this microflora, then at that point we have a baseline set up, and if something invasive then enters the system, then we can go back and look and maybe figure out ways to keep this from happening in other environments, or maybe be able to kind of fix what's going on there. Also, it's a good environment for microbial ecology because there's no light penetration when you start getting back into the cave. And if you think about it, this is the most easily accessible environment that I know of where you can get that lack of light penetration. And also, there's a fairly low nutrient influx. Without the light, there's the lack of photosynthesis causing your primary production. So this really is an environment that, like I said, I mean, I don't know how you could get to an environment like this as easily as you can with the caves, and it makes for a pretty unique setup. Also, these are traditionally thought of to have less complex biofilms. I'll let y'all make y'all's judgment calls after you look at the data, but I say that could be a little bit off. It's based on older techniques, and I'll go into more detail on that. And these newer techniques are showing some interesting things going on here. And there's also some possible applications that can be brought up from looking at this bacteria and things and that being this industrial. And I'll go into more detail on all that as well today. So the site chosen for this is Blowing Spring Cave, and it's located in northwestern Alabama, and it's a forever wildland tract through the Department of Conservation and Natural Resources. And it's a restricted area, and it's so there's limited access. And that's to preserve this habitat for this listed species of bat, the gray bat. All right, the ecology of a cave, like I said, is pretty unique, and caves can be formed through a lot of different ways. You can have lava flows that cause cave formations. You can have water erosion causing cave formations. Various ways. But typically speaking, it's through the flow of water and through carbonic acid, at least in the region that we're at here with limestone. And that's how this particular cave is made. There are several zones within the cave. There's the entrance zone in which you walk into this area. You can see a little bit of light. You know, if you're going in with your helmet light, you know, turn off your light on all your lights, you can still see a little bit in the entrance, but it's getting pretty dark. You extend into what they call the twilight zone. No jokes. And this area is really where you start to see non-cave species start to phase out. Towards the entrance and into that twilight zone, you may see a lot of salamanders, like slimy salamanders, cave salamanders, stuff like that. Once you break that line and going into that twilight zone, it's almost like there's an invisible barrier that keeps organisms from going back through there. After you cross through that particular zone, it's almost specific to the cave species that are have come to adapt to that particular environment. And like I said, hydrology is not only important in the formation of these caves, which is one of the biggest players in it, it's also important in nutrient flow. If you have, say, a creek flowing through the cave like we do in this cave, then that's going to be bringing in nutrients and organisms from the outside environment and bringing them in through the system. And it can also flush out the system as well if there's a flood or something like that. So that's a very important player in the nutrient flow as well. In nutrient flow in general, you have organisms such as bats that live in these caves, and you know, they're doing their thing. You have you know, guano fecal matter from the bats. You have you know, if there's bats that die and drop down onto the floor of the cave, that's another source of nutrients, because you have to think, again, there's not primary production here, there's not plants here creating a carbon source for these organisms. So, like I said, again, it's traditionally considered to have low biotic composition in these cave systems because of this kind of limited nutrient flow and the lack of light. These organisms have kind of developed a maintenance over growth type situation where, for example, say you streak a plate with something like E. coli in the lab, you can have that plate grow up overnight with, you know, several hundreds to several thousand, you know, 1,000 colonies. And with these samples like this, you might end up with a growth rate that's more like you might get a colony every month or up to a year. I mean, they're very slow going. And also, there's been a lot of evidence to support that there's hemolithotrophic activity in these caves, which is pretty interesting. Limestone itself contains a lot of inorganics that can be used through redox reactions, like magnesium and sulfur, things like that. And this is sort of the possibly a way that they're mediating primary production in these systems is through these hemolithotrophic activities, since there's photosynthesis. So there's also, like I said, stuff like the backbone and things like that. And with that, you're getting chitin in the system, whichever way you want to say it. And this is a really difficult substance to break down. And it's also a big problem with the seafood industry of the waste of it, because it can contain up to about 40% of what they're getting is actually chitinogenic waste versus what we're actually eating. So there's a lot of waste there. And it has a lot of potential to be used for things beneficial, such as I've read that it can be used as, after being broken down by these bacteria, if you find some bacteria that can break it down, then it can then be used as filler for food, for livestock and things like that. So it does have a lot of potential industrial applications. If you think about the amount of tonnage, I think they said it was like I said, 50% of the crustaceans, things like that, in the seafood industry. So that's a lot of waste there. Also, to make these systems work with these low nutrients, we might be finding some pretty interesting complex chemical chains going on. Organisms that are taking one form of, say, nitrogen, let's just take, for example, ammonia and turn it into nitrite, and then maybe another organism uses that nitrite, and then eventually it's turned back into ammonia and brought back around for the other organisms to use. So there could be some pretty complex chains of events going on there. Also, there's a very limited supply of nutrients relative to some other environments, so there could be some pretty unique microbial competition, which could also help us lead to some of those medicinal applications, such as antibiotics.